viewers. My name is Lusanda Precious Vika from Department of Education, a subject advisor in OR Tambo Inland. Welcome to the ECDOE broadcast studio. Today's lesson will be covering the topic for your full understanding. Today's topic, I will be talking about animal nutrition. I will be explaining about the comparison between ruminants and non-ruminants, their functions, the external structure, the internal structure, the enzymes that are in it, and we'll end up talking about all the calculations that we have in agricultural science. Now, we have a fowl and a pig. These are the non-ruminants. As you know that we have classified our farm animals into two, the ruminants and the non-ruminants. We know that protein, once it is in an animal's feed, we know that the animal has grazed the protein from the pastures. The source of protein is nitrogen. And through the life cycle of in nitrogen from our previous grade, the grade 11, we know how does the soil, the plants gets nitrogen through lightning and rain. So the source of protein is nitrogen. Also, lastly, there should be continual elimination of end products. The end products I have just mentioned, that if fatty acids, they should be eliminated by being absorbed directly into the walls of a rumen, and the gases and carbon dioxide should continuously be expelled through the mouth of an animal during belching. Okay, the last two compartments. The first two is rumen and reticulum, and I combined them as they perform more or less the same function. And then the animal regurgitates the food back into the mouth during the process called rumination. The feed or the grass or the food gets into the mouth of an animal, into the rumen and the reticulum, and then the animal regurgitates it into the mouth where it is re-chewed. I said it partially chews the feed. And then when it regurgitates back into the mouth, it chews it. That is the physical digestion. And then re-swallows it, meaning it swallows it for the second time. It chews it for the second time. And then it mixes well with the saliva in the mouth of an animal, straight down into the omasum. And when it reaches the omasum, it is where the undigested feed particles are moved back into the reticulorumen together with excessive amounts of water. And in the omasum, there is removal of water and the drying of feed and the grinding of feed. And at the same time as these leaf-like layers in the omasum, they push the food into the abomasum whilst bringing back the undigested feed particles into the rumen and reticulum as the structure you see in the diagram. So it is a leaf stomach. These leaves, they act well with each other, pushing the food into the abomasum and removing excessive amounts of water back into the reticulorumen. So here it says an omasum, leaf-like folds that grinds food particles prevents the coarse food from entering into the abomasum, dries food by retaining back water into the reticulorumen. Also, as you can see, these pimple-like projections. Here, the papillae are these pimple-like projections where I said they appear the most in the rumen. So the volatile fatty acids, that is the end products of a volatile fatty acid, you remember, I said it is the acetic acid, the butyric acid, and the propionic acid. They are absorbed there. And then the last part of the stomach is the abomasum. This is the true stomach, the milk stomach, as I have mentioned before, in the case of that young ruminant animal. It is where secretions are taking place. I talked about the secretions in the stomach of a farm animal. In the stomach of a farm animal, these are the gastric juice and the iusis. They are the same also as in a cow. Now, let us get into the small intestine. The small intestine is from when food comes out of the stomach. It gets into the small intestine. Remember, food from the small intestine, the pH medium is acidic because of the action of hydrochloric acid. And the medium in the small intestine is alkalinic. So each food that is released, that is, we talk of chyme, not food. We talk of chyme. When the chyme gets out of the stomach into the small intestine, there is a secretion that is made by a liver called a bile. It immediately changes the pH from acidic to alkaline. And also the secretion that is made by the pancreas. It changes the pH from acid to alkaline. So meaning both the liver secreting the bile changes the acid medium to alkaline medium. And also the secretion made by the pancreas, that is the pancreatic juice, it changes the pH from acid to alkaline. And then what does the liver do? The liver secretes the bile. Bile is not an enzyme. It is just a substance. So when we talk of bile, we don't talk of an enzyme. Bile is contained by a gallbladder in the liver, that greenish, alkalinic, sour substance. So what does it do? A bile breaks down fats, okanye hydrolyzes fats, okanye breaks fats into tiny droplets. Also, bile secretions are antiseptic. The secretion that is made by bile is antiseptic. Bile promotes the absorption of fatty acids and glycerol. Also, it assists in the absorption of fat-soluble vitamins, vitamin A, D, E, K, fat-soluble vitamins, that is the ADEK. And then the pancreas. The pancreas secretes the enzymes. 3 enzymes are secreted by the pancreas. The enzyme that the pancreas secretes it digests the three components of a feed. Fats are digested by these enzymes. Protein is digested by these enzymes. Carbohydrates is digested by these enzymes. Now let us look at the enzymes secreted by the pancreas, the trypsin. The trypsin digests a protein into peptones. Lipase, it digests fats into fatty acids and glycerol. Amylase, it digests the carbohydrates. That is, it digests a starch into a maltose. Then another secretion in the small intestine is by the duodenal gland, the succus entericus. Succus entericus has got an enzyme, enterokinase. The enzyme enterokinase, it activates the inactive trypsinogen into trypsin. And then trypsin digests the peptones into peptides. That is, the proteins are being digested there by a trypsin. And then a range of carbohydrates, that is, the maltase, the sucrase, the lactase, digest carbohydrates into their simplest form. We have a lactase that digests lactose into glucose. We have sucrase enzyme that digests sucrose into glucose and fructose. We have lactase enzyme that digests lactose into glucose and galactose. I think I've made a mistake. A maltase enzyme digests maltose into glucose. Then a lactase enzyme digests lactose into glucose and galactose. Here's an activity. We have to work it out. Name the farm animal presented by the alimentary canal in the diagram above. Justify two reasons which are visible in the diagram above to support the answer to question 2.1. When a question reads, justify two which are visible, so you have to give visible reasons, not the reasons you know, just visible reasons. And then 3, indicate the pH of the contents of each of the following parts. The contents of part B and the contents of part C. 4, state 2 important digestive functions of substances secreted in part B. Importance of digestive functions secreted in part B. And then 5, explain how part C is adapted to perform its function. And then here we go. Let us look at the answers. The farm animal in the picture is a fowl, chicken, or poultry. Remember, Mfundi, the question reads, identify or name. When the question says name, you give the name of the animal. But when the question says classify, you give the class of an animal, that is the ruminant or the non-ruminant. So, Mfundi, you will not get marks if you say non-ruminant fowl, because what is needed here is the name of farm animal. And then a question about justification by what is visible in the picture. It has got a simple stomach, okanye monogastric stomach. The presence of gizzard or ventriculus and ventriculus. The presence of ceca, that is the two blind spots. It shows that it is a fowl. And a presence of a crop. This part is a crop. So it gives you full marks because we have indicated the presence of ceca, the presence of a crop, and also the presence of the ventriculus. And then a question indicates the pH contents of each of the following parts. Here, this is the proventriculus, the medium for a proventriculus, that is the stomach. The medium is obviously acidic. And in the small intestine, the medium is alkaline, okanye basic. The importance of digestive functions of the substance secreted by B, that is the gastrics. It is antiseptic and it destroys bacteria, that is that hydrochloric acid. It prevents the rotting of the contents of the stomach. It changes the alkalinic medium into acidic medium. Disaccharides are changed into monosaccharides. Activates, that is the functions of hydrochloric acid. It activates the inactive pepsinogen into active pepsin. Pepsin changes the proteins into peptones. Then the role played by part C. The thick muscles with stones to grind food. Remember, we said it is where physical digestion, mechanical digestion takes place. Now, the calculations. We have 5 calculations. The coefficient of digestibility, the nutritive ratio, the Pearson square, the fodder flow program. You have to ensure that when you're doing these calculations, there are implications. You have to interpret the calculation. You must know why a fodder flow, why a nutritive ratio, why a digestibility coefficient. So let us look at the digestibility coefficient. Digestibility of a feed first is that portion of a feed that is ingested by an animal, digested, absorbed and used by the body, and not excreted. So that feed, we count it as it is digested. So the composition that affects the digestibility of a feed, it is the composition of a feed. What does the feed have? The composition of a ration. What does the ration have? The preparation of a feed. How the feed is prepared. The type of an animal. Is this animal that we have prepared feed for a ruminant or a non-ruminant? Because if you prepare a feed for a non-ruminant, then you give a feed for a ruminant, it means that feed won't be digested. Then the quantity of feed consumed. It means the more of feed that is being consumed, it will have less digestibility. We know when we eat a lot of food, it means the digestibility will be low. And then the age of a plant. If the plant is old enough, it won't be easy for an animal to digest. So when calculating a digestibility of a feed, we're working with a dry matter because feed has got a moisture part and a dry matter part. So if a feed given to check the digestibility has got moisture in it, you have to first remove water. Then we have the formula for digestibility of a feed. I want to emphasize this, good learners, that when we calculate digestibility of a feed, we work with a dry matter, and it is expressed in a percentage. So this is a formula. Digestibility coefficient is equal to dry matter intake in kgs and dry matter manure in kgs divide by dry matter intake all times 100. So I said it is in a form of a percentage, and we're working with a dry matter. Now, here's an activity. A cow eats 30 kg of concentrates with a moisture content of 10%, and 16 kg material is excreted with the moisture, excreted in the manure with a moisture content of 35%. Remember, I said we're working with dry matter. So this is the calculation. We have to first remove water. Another shortest form of using water, of removing water, we say 90 over 100, because learners used to forget to minus the 3 kg from the 30 kg to work with 27 kg. When they get into this 3 kg, they come take this 3 kg into the dry matter intake. Whereas, remembering that this 3 kg is the kg of moisture that needs to be removed from the feed. So I used to tell my learners that you first minus the percentage here. So 10 minus 100 minus 10 is 90. So you work with 90. Say 90 divided by 100 times 30. So the easiest way to remove water, because you used to forget to minus this 3 kg from the feed, the actual feed, which is 30 kg. So you rather minus, you rather work with a difference of 10 to 100, which is 90 divided by 100 times 30 is equal to 27. It gets straight to the dry matter content. And then the 35 moisture content. So 35 divided by 100. So you minus you get the difference of 35 to 100, and is 65. 65 divided by 100 times 16 kg. You get straight into the dry matter. So that is a mark. And then we substitute that. And then we do the substitution, the dry matter intake, dry matter manure, divide by dry matter intake times 100. We do the substitution. And remember, we said we're working with dry matter, and presented, the calculation is presented in a percentage. I don't know. Presented in a percentage. So there is an implication. We have to interpret the calculation. That, let us look at the difference now. A feed of 100, only 61.4 has been digested and absorbed by an animal, meaning 38.6% is the part of the feed that was not digested, was not absorbed, and was excreted by an animal, which is a loss for umfama at the end of the day. As of now, let us stop here. We have come to an end of our lesson. You can leave your questions on the comments. Don't forget to leave the name of your school so we can give shout out. Please share, recommend, and subscribe to our YouTube channel. Thank you for watching. See you on our next lesson. Bye-bye.